Hello students, how are you all? I hope everybody is fine. See, today we are going to start with a new chapter and a very interesting chapter, that is biotechnology principle and then process. Okay, now biotechnology principle and process. This comes under unit ninth unit. Okay, biotechnology, it comes under unit ninth unit, children. This biotechnology principle and process is very unique chapter, I can say, because something new, whatever the new inventions this era is done, no, that is the biotechnology. Much more progress is there in biotechnology, much more discoveries were happened in biotechnology field. So that let us understand what actually the concepts, and then what are the principles are there, which kind of process are going to be takes place in biotechnology. Before going to that, I would like to tell you the definition of biotechnology, children, what actually it is means. Okay, it is a branch, it deals with the technique of using live microorganisms or any of the enzymes from organism to produce the products and process useful to the human being. Biotechnology process is finally, it is useful to the human beings only, but for the human beings use, we are going to use the microbes and also the enzymes. Okay, now let me write the definition. Definition of this is: the biotechnology deals with techniques of using live microorganisms or else enzymes from the organism to produce what product, to produce product. And this process is useful to whom? This process is useful to human, it is useful to human being. Okay, and the third term, see, it deals with what? Technique of using live microorganisms. Here we are going to use live microorganisms genome. In specifically, I can say the genome. Already you people know about DNA, genome, gene, the structure, everything you learnt in molecular basis. So I think, I hope there is no necessary of explanation those all things. See here, either we may go use of live microorganism or else we may go use of enzyme, but in welfare of whom? In welfare of human being. The term biotechnology was coined by the term biotechnology. The term biotechnology was coined by Carl Ericke. Carl Ericke in 1917. Okay, now in 1917, many of the process will comes under biotechnology. Those process many techniques are also included under biotechnology. What are they? Examples I can give you: in vitro fertilization, in vitro fertilization, and then synthesizing a gene and using of it. Many of the different genes are going to be used, and developing a DNA vaccine and correcting a defected gene. And many of the corrections, like if any of the gene is defected, then what happens, that defected gene is going to be corrected by using what, children? Biotechnology. This is one of the very major technology and then very useful technology, I can say. And then according to the European Federation of Biotechnology has given a definition of biotechnology. See, for a MCQ, this definition may useful. Who gave the next definition? European Federation of Biotechnology. European Federation of Biotechnology, that is EFB. Expand EFB means European Federation of Biotechnology has given a definition of biotechnology. Children, let me write that one. What they said: the integration of natural science, the integration of natural science and the organisms, cells, parts thereof, and molecular analogue for what? For the products and services. This is what actually the European, that is EFB, is giving. The integration of natural science means the elimination of natural science in what? In organism cell, or else part of cell, or else molecular analogue means in molecular level we are analyzing the molecular level like DNA, RNA, ribosome. This molecular level we are going to be manipulate them. That one means a molecular analog. For what? For the product means like antibiotics, many of the vaccines, and then many of the resultants, okay, and then many of the services. Now let us talk about principles of biotechnology. There are mainly two main techniques of modern biotechnology, that is genetic engineering and then chemical engineering. We are going to discuss in deep about that. What actually this? How many are there? There are mainly two main techniques are there, children. Two main techniques. First one, genetic engineering. I think you people already heard many of the times about the genetic engineering, what actually it is. What is this genetic engineering means? It includes techniques to alter the nature of the genetic material. That's what it is called, genetic engineering. What? It includes techniques to alter the nature of what? Genetic material. What are the genetic material? In whole the world there are only two genetic materials, that is DNA and then RNA. Okay, now if you are going to alter the whatever the information is there in the DNA and then RNA, that alteration of DNA and then RNA sequence by using technique, then it is called what? Genetic engineering. And the second one is chemical engineering. What is chemical engineering, children? It includes maintenance of sterile microbes, contamination to enable the growth of only desirable microbes in the large quantities for the manufacture of biotech products like antibiotics and then vaccine and then enzyme. Here what happens in chemical engineering, it involves what? Maintenance. It involves maintenance of sterility. Sterile microbes means like only useful microbes we are going to maintain. If useful microbes are contaminated with any other microbes, there is no use. Okay, so in chemical engineering, maintenance of only the pure microbe is very important, and then we are going to avoid or else contamination to enable the growth of any only desirable microbes in large quantity. Only desirable microbes are going to be grown in large quantity. Okay, in this what happens? Only desired microbes are going to be grown in large quantities for what? For manufacture of, for what? For manufacture of biotech products like what, children? Antibiotics, that is penicillin and all, and then most of the vaccines and then enzymes. Okay, these are very helpful for in pharma industry also. There is much more use of these two area, that is genetic engineering also very useful. Why? Because it is by using of genetic engineering only we can manipulate many of the hereditary disorders, like from if any of the disease is from hereditary, like one generation to the another generation any of the disease is going to be formed, that kind of hereditary diseases can be maintained by using genetic engineering. By using chemical engineering, many of the vaccines are going to be produced. Okay, now what happens? The conceptual development of principles of genetic engineering we are going to like, means what actually the concept, what actually development is that. Okay, now only introductory part we are going to learn today. Okay, conceptual development of principles of genetic engineering. Okay, see children, this chapter itself is the main basic chapter. Next, whatever the chapter is there, no, that is biotechnology and its applications. In applications you are going to learn how actually the biotechnology is going to be applied. In this chapter itself says what actually it is says, biotechnology principle and process means what? Only the principles we are going to learn. Okay, sorry, I forgot to say this chapter carries 6 marks, children. Okay, 6 to 7 marks, 5 plus 2, 5 marks will be compulsory, and then 2 marks will be, or else 1 plus 1 you can expect from this. Okay, now genetic engineering is a kind of biotechnology which deals with manipulation of genetic material by man in vitro condition. Means what? See, the biotechnology is what? It is a kind of technology, children, where manipulation of genetic material like DNA or else RNA is made by man in vitro condition, means in laboratory condition. In vitro means what? In laboratory condition. Genetic engineering is based on two important discoveries in bacteria. Genetic engineering, this genetic engineering is based on, genetic engineering is based on two important discoveries which turned the world itself, children. That is in bacteria, that is presence of plasmid in bacteria. This is very 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 important discovery, I can say. Okay na? Where what happens, children, which can undergo replication, which can undergo replication along with and independent of chromosomal DNA. Means what? If this is a bacteria, okay, bacterial, this is genomic chromosome DNA. Along with that, there will be a circular DNA which is called extracircular DNA, is called what? Plasmid. This is what, children? Plasmid. Okay now, here what happens, this is one of the best discovery, I can say, in the bacteria. Here, when the bacteria is undergoing for the replication, multiplication of bacteria takes place, this plasmid has the capacity to replicate independently, and also it has the capacity to replicate along with the chromosome of the DNA. Means what? Just think, if we are going to add any of the DNA to this, any of the alien DNA to this, what happens? This plasmid is going to be replicated. Later on you are going to learn about that, don't worry. And then second discovery is restriction endonuclease. Okay, this is enzyme, children, which can break DNA at a specific point. At what? At a specific point. So they are called what? Molecular scissors. The restriction endonuclease are called what? Molecular scissors. Okay now, why they are called molecular scissors? Because if I wanted to cut the DNA at this specific point, I can use a specific restriction endonuclease that will go and then cut that DNA. Why we are going to cut that DNA? If that cut is there, then only you are going to insert. No, that's what. Okay, I hope you people are going to understand this. In 1972, genetic engineering was started by Paul Berg. Where? In 1972. Who started? In 1972, genetic engineering was started by Paul Berg. Okay, and then was able to introduce a gene of, he was able to introduce a gene. A gene means a part of the DNA of SV, name that is SV40 virus into what? Into a bacterium with the help of a vector, with the help of what? Lambda phase. What? With the help of what? Lambda phage. Okay na, from the virus to the bacteria, he is going to introduce what? SV40 gene by the use of what? Lambda phase. Okay na, lambda phase is nothing but again a virus. So the Paul Berg is called what? Father of genetic engineering. Who is called the father of genetic engineering? Paul Berg. The technique of genetic engineering include, there are many of the steps are there. The important things is formation of recombinant DNA, that is rDNA. rDNA means recombinant DNA. Recombinant, combination of two genes or else two DNA. Second one, using of gene cloning. Means the gene which is recombinant, it is cloned many times, multiplied many times. Third one, gene transfer. Gene is transferred, which is very desirable. It permits to isolate and introduce only one or set of desirable gene. For a bacteria, you can introduce only one gene, or a set of the gene also you can introduce. A piece of DNA which is introduced into alien organism would not be able to multiply itself in the organism. For example, I can say, if I introduce a piece of, see, this is bacteria, this is genome, and then this is plasmid. Many of the students will get doubt that why can't I introduce a DNA in the cytoplasm of the bacteria. See, when bacteria is introduced with the DNA, then what happens, this bacteria thinks that this is alien material. So from the bacterial body, what happens, there will be release of enzymes which will break down this DNA. So whenever you are going to introduce this kind of DNAs to the organism, it will fails off. But instead of doing so, you can introduce the DNA to the plasmid. When you are going to cut this plasmid at a specific point, that specific point is called origin site. Okay, at the origin site, by using restriction enzyme, if you cut, means it will just gets open, and then you can introduce your alien DNA. So that what happens, when this plasmid is going to be multiplied, that DNA itself is also going to be multiplied. But when it gets incorporated into the genetic material of recipient, now this is recipient, it may multiply and then be inherited along with the host DNA, as alien piece of the DNA has become part of the chromosome, which possesses the ability to replicate. These words are given in your NCERT, just I am going to read it out. Okay, there is a specific DNA sequence called what? Origin of replication, or else origin site. Children, origin of replication also you can say, that is responsible for the initiation of what? Replication. Which is responsible for the initiation? Origin site, or else origin of replication. An alien DNA can be linked with the origin of replication, can be replicated and then multiply itself in host organism. It is called what? Cloning. And this host organism, how many times it is going to be multiplied? No, that the alien DNA, now this is what? Alien DNA. Okay, this alien DNA is also going to be multiplied that much time. Okay, now forming the multiple identical copies of any template DNA is called what? Cloning. Cloning means single ಇರೋದಕ್ಕಿಂತ better 100 copies, 1000 copies, lakhs together copies is better. Why? Because if only one multiplication, only one gene is going to be multiplied, only one protein is going to form. If the same gene is going to multiply in millions together form, quantity is more, means the productivity will be more. That's what the cloning is also very important in this manner. Okay, now, so for today we are just in the introductory part, children. From the next I am going to explain you in the deeper manner. Okay, thank you.